Good morning, students. Welcome back to biology class, chapter 8, Cell, the unit of life. When you look around, you see both living and non-living things. The living things are composed of cells. Some are composed of a single cell, that is called unicellular organisms. Others are composed with many cells that are called multicellular organisms. Okay, what is a cell? Cell is the fundamental structural and functional unit of all living organisms. Okay, unicellular organisms are capable of independent existence and performing the essential functions of life. Anton von Leeuwenhoek first described the living cell, and he observed also, but Robert Hooke discovered the cell. There are two types of cells, prokaryotic cells and the eukaryotic cells. In 1838, Matthias Sheldon, a German botanist, observed that all plants are composed of different kinds of cells. Okay, the invention of microscope revealed the structural details of the cell. After that, Theodore Schwann, 1839, a British zoologist, studied different types of animal cells. Okay, after that they find out a thin outer layer which is called plasma membrane in the animal cells. He also concluded that based on his studies on plant tissues that the presence of cell wall is a unique character of the plant cells. On the basis of this, Schwann proposed the hypothesis that the bodies of animals and plants are composed of cells and the products of cells. Sheldon and Schwann together formulated the cell theory. This theory did not explain how new cells are formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells, that is called omnicellular cellula. He modified the hypothesis of Sheldon and Schwann to give the cell theory a final shape. Cell theory concludes, understood today, it is like this. First point, all living organisms are composed of cells and products of cells. All cells arising from pre-existing cells, that is the second point. Now we will discuss about an overview of cell. All cells contain cytoplasm and ribosomes. First we will discuss about the cytoplasm. A semi-fluid matrix where cellular activities and chemical reactions occur. This keeps cells in living state. After that, ribosomes, non-membrane bound organelles found in cytoplasm, chloroplast, mitochondria and on rough endoplasmic reticulum. Beside the nucleus, the eukaryotic cells have other membrane-bound distinct structures called organelles. That are the examples: endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane and bound organelles. Okay, ribosomes are present in both the cells in the eukaryotic and prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also with the two organelles, chloroplast and mitochondria and rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle is called centriole, which helps in cell division. Cells differ greatly in size, shape, and activities. For example, the smallest cell, that is the mycoplasma, it contains 0.3 micrometer in length. Largest isolated cell is eggs of ostrich. Longest cell is nerve cell. Size of bacteria 3 to 5 micrometer. Human red blood cells 7.30 micrometer in diameter. Based on the functions, cells, shape of the cells may be disc-like or polygonal, columnar, cuboid, thread-like, or irregular in shape. The cells do not have a particular shape. Based on the function, they are divided into these all the forms. Next, we will discuss about the prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO, pneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cells. They may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, that is rod-like, coccus, spherical, vibrio, comma-shaped, and spirillum, spiral-shaped. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not developed, enveloped by a nuclear membrane. See, that is the main point. The genetic material is not enveloped by a nuclear membrane. In addition to the genomic DNA, a single chromosome, circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. Okay, bacteria have the extra chromosome, that is called the plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is the resistance to the antibiotics. Okay, next, DNA is used to monitor bacterial transformation with the foreign DNA, that is using in the biotechnology. Nuclear membrane is bound in eukaryotes. No organelles like the ones in eukaryotes are found in prokaryote cells except for ribosomes. Prokaryotes have something unique in the form of inclusions. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. They are essentially enfoldings of cell membrane. Okay, next, each part we will discuss. First, cell envelope and its modifications. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure. Okay, three-layered cell envelope consists of three-layered structure. The outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Although each layer of the envelope performs distinct function. Okay, glycocalyx, cell wall, and the plasma membrane are the three-layered structure. They act together as a single protective unit. Bacteria can be classified into two groups on the basis of the differences in the cell envelopes and the manner in which they respond to the staining procedure. developed by the scientist Gram. Those that take up the gram stain are gram positive, and the others that do not are called gram negative bacteria. When the staining time, they are absorbing the color, they are the gram positive, and that other bacteria are the gram negative. Glycocalyx, that is the outermost layer, differs in composition and thickness among different bacteria. It could be loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. Okay, the bacteria outer layer is called the glycocalyx, that in the some of the bacteria it is called capsule. The cell wall determines the shape of the cell and provides a strong structural support to prevent the bacterium from bursting and collapsing. Because of that only they can live in the extreme conditions. The plasma membrane is semi-permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extensions of plasma membrane into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in the cell wall formation. What are the functions? They help in cell wall formation, DNA replication, and distribution to daughter cells. They also help in respiration, secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores coloring, which contain the pigments. Chroma means coloring, pigments. Bacterial cells may be motile or non-motile. Movement. If motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangement of flagella. Bacterial flagellum is composed of three parts. First one, filament, hook, and basal body. The filament is the longest portion and extends from the cell surface to the outside. Besides flagella, Pili and Fimbriae are also surface structures of the bacteria but do not play a role in motility. The pili are elongated tubular structure made up of a special protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Next, we will discuss about the ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made up of two subunits, 50S and 30S units. When present together form 70S prokaryotic ribosomes. S or Svedberg's unit stands for the sedimentation coefficient. It indirectly is a measure of density and size of ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes or polysome. The ribosomes of a polysome translate the mRNA into proteins. Next, inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. These are not bounded by any membrane system and lie free in the cytoplasm. Example, phosphate granules, synophysian granules, and glycogen granules. Gas vacuoles are found in the blue-green and purple and green photosynthetic bacteria. Thank you.